So first off, I'll take you to a little overview of the structure of glycogen. So here we have our glycogen here, okay? In the middle of our glycogen, we have a protein called glycogenin. Okay, and this is really, really important. I'll explain to that why. It might surprise you that there's a protein in the middle of a glycogen, of a carbohydrate store, but there's a very good reason for this, and we'll get onto that probably in the next video. Okay, so what we have is we have a combination of these glucose molecules, which aren't actually glucose molecules, but what we talked earlier, they're called glucose residues, okay, because they've got phosphate groups bound to them, which energizes them and helps them stick together. Again, we'll talk about that a little bit more in the next video when we talk about glycogenesis, the building of that, because it's a slightly more complex process. So we'll start with the simpler one first. So we have these chains of these glucose residues here that come together to form glycogen. Now this is obviously a two-dimensional reconstruction. We'd also have glycogen molecules coming off in this direction as well. So this is a big three-dimensional structure that makes up what is called a glycogen granule, which, like I said before, sits inside our cells in the cytoplasm of cells, or the sarcoplasm if you're talking about muscle cells, and is there to provide that energy in close proximity. Okay. A few things to note, which I'll discuss in a bit more depth in a minute, is to looking at the spacing between these. So when these chains are placed onto this, which we'll talk about how that happens in the next video, like I said, there's got to be a certain amount of spacing between this, and the reason for that will become clear very shortly. So the types of bonds that occur between these glucose residues are called alpha-1,4 glycosidic bonds. So they're the ones that combine in a long chain. There's also a second type of bond called an alpha-1,6, I don't know why I haven't written that, glycosidic bond, and they're responsible for creating these chains. Now you might just think that the process of breaking those off will be the same, but they're different, and I'll explain to you why now. Okay, so let's say we start at the end of the chain here. So I'm going to take these 1, 2, 3, 4. I'm going to take these 5 glucose residues. I'm just going to put them here. Okay, so they would still be attached. Okay, and they're broken down by a process called phosphorylasis. It's a similar process to hydrolysis, which is when you use water to break things down, except instead of hydrogen, it donates phosphate. So what we get is so if we had these 5 here sat on the end, we'd get one breaking off the end, like so, which would leave the 4 here. It donates a hydrogen ion to it as well, okay? And then we get it's a glucose molecule but with a phosphate attached to it, and that is known as glucose 1-phosphate, okay? So it's glucose 1-phosphate. Glucose 1-phosphate is not usable in the body, okay? So it is converted into glucose 6-phosphate. Glucose 6-phosphate then enters down 10 more steps to make pyruvate, which can then be used to make ATP. At pyruvate, it then either goes on to create aerobic glycolysis, so the breakdown of glucose for aerobic glycolysis, or if there's no oxygen present, we have anaerobic glycolysis, which then gets turned to lactic acid, which then re-enters back into, which then can be stored as glycogen again through gluconeogenesis, and then back in through the storage way when we talk about synthesis in the next video. So the enzyme that does this, unsurprisingly, is called glycogen phosphorylase. Okay, so I'll write that out anyway. You don't need to remember this, but it's just glycogen phosphorylase, and that is, like I say, is responsible for breaking down all these, and it will then go on and remove these chains until we end up with a whole host of these G1Ps to be converted to these G6Ps here, okay? The problem comes, let me just take this off for now because it's not important. So we know how to make all these ones, okay? So like you can see, glycogen isn't actually technically broken down into glucose. It's broken down into glucose 1-phosphate and then it's converted to glucose 6-phosphate. The problem becomes when we've removed these here, so let's say we've done all these, like so, we will get to a point where our glycogen phosphorylase, it cannot act on this. There's not enough, how would you describe it? There's not enough space on the enzyme, or there's not enough space in the enzyme to actually bind onto this. So another enzyme comes in, which is called the branching enzyme. branching enzyme. And this consists of two parts. Part one is what is called a transferase. So I'll just put trans there for now. And what transferase does is it comes and it takes these first three here and it sticks them onto the end of another chain. Still got those bonds in there, so I'll just draw the bonds on this one for now. So our phosphate bonds in here. Okay, well what you can see now is so then that can be acted upon via glycogen phosphorylase to then reduce these back down to your glucose 1-phosphate to be converted to glucose 6 again to then enter ATP. What happens with this one here is there's a second part which is called glucosidase, I think. Glucosidase? Trying off the top of my head. I think it's called glucosidase. And whereas these reactions here and the trans and the whereas the glycogen phosphorylase acts through phosphorylasis, this acts through hydrolysis, okay? And because of that, we actually get, instead of getting a glucose 1, we actually just get a molecule of glucose. What this means is that when we have a glycogen, basically 90% of what we get from glycogen is G1P, and only 10% is glucose. It's just our G. Okay, God, my writing's awful today. I apologise. So just 10%. So when you break down glycogen, you only get 10% glucose. That's not a problem because, like I said before, glucose itself needs to be converted again to this G6P through a different through another enzyme which phosphorylates it. So then G6P can then enter the cycle again. So that is basically how this takes place. This one gets removed, leaves us with our glucose here, and then a whole host of glucose 1s as well. Okay, so that is how glycogen is broken down to then enter into our metabolic pathways for glycolysis, and then eventually into the Krebs cycle for use to create ATP. So that's how we break down glycogen. The next one we're going to look at then is how we build it up, because it's a slightly more complicated process, but only slightly. But I think it should be obvious to see from now how if it's built up one way, if it's broken down one way, it's going to be built up in the exact opposite way. It just uses a slightly different process. So give you guys a few minutes to digest that, and I will be back very soon. Okay then guys, in the last video we talked about glycogenolysis, so the breakdown of glycogen. So now we're going to move on to glycogenesis and how we create glycogen for storage. Okay, like I say, this is a little bit more complex, but not so complex. Like you see, it does pretty much mirror glycogenolysis. So we start with our lonely molecule of glucose, or what is actually known as, in the scientific terms, as alpha D glucose. That's the stuff we get from our food. Okay, that's floating around, and a special enzyme called glucokinase, or GK here. Don't worry too much about this terminology. It's fine. You'll find this stuff if you look online anyway, so it should be easy enough for you to brush up on if you want to. Converts this to glucose 6-phosphate, familiar one that we looked at before, which then can go down to perform glycolysis to produce energy. Okay, so it's the same pathway we went the other direction before through glycogenolysis that we got to this G6P that produces glycolysis for ATP through either the anaerobic glycolysis system or the aerobic glycolysis system. Okay, so we also then, so we've got this G6P floating around. In order to get this to convert to eventually to storage glycogen, we need to convert it to glucose 1-phosphate, and this is done by an enzyme called phosphoglucomutase. That was a struggle for me to remember. So phosphoglucomutase, and that works both ways as well. So like I said before, the pathway works both ways, so it can convert it back. We then have what is we call a nucleotide. It's a nucleotide with some phosphates attached to it, which is called uridine triphosphate. Okay, so an enzymatic reaction happens here, and we end up with what is called UDP, which is uridine diphosphoglucose, and we end up with some inorganic phosphate atoms floating around to do whatever. Okay, it's this UDP is a really important step in this, because the UDP has still got the fundamental structure of glucose, but the extra phosphates do two things. Firstly, with the action of glycogen synthase, it can then combine to our chain of these glycosyl residues here. I keep calling them glucose residues, they're actually glycosyl residues, okay? So glycogen synthase does that, but UDP also, because the extra phosphate provides energy as like a primer, so it activates the glucose in conjunction with glycogenin, which is the protein in the middle, okay, in order to create this primer, which then allows the combination with the glycogen synthase to attach this glucosyl residue to our chain of other glycosyl residues to increase this in length. Okay, so that's how we build glycogen. Just give me 1 second while I just get my rag here, because we're going to talk about the final step in this. So if you remember last time we talked about how then we have debranching enzyme. Well, the opposite to debranching enzyme is branching enzyme. So why would we need this? Well, first and foremost, let me just move this out of the way. Not that I need this much space. Okay, first and foremost, if this is our granule of glycogen, there's only so big this can get. So if we imagine we've got a limit on the size of our granule that goes around like this and continues around and so on and so forth, there's only so far we can get adding these glycosyl residues till we can get to the end. So how do we get them to fill in the space and to grow out in this direction as well towards you? Okay, well, we have a thing called branching enzyme. Branching enzyme, what that does is it takes, in the opposite way of debranching enzyme, it takes a number of these residues, separates them off, 6, 7, there's a reason I'm counting 7, I'll explain in a minute. So it takes those 7 off, there, and it will place them somewhere along this chain here. Now there has to be, remember, 1, 2, 3, at least 4 spaces before it can then place this chain of 8. No, it's just broken off. 1, 2, 3, 4, 5, 6, 7. So it's 8 in that instance, okay? The reason for that is obviously because we've got the enzyme action on the way back with the debranching enzyme that needs a certain amount of space to be able to operate, to remove those 3 that we said, and then attack that final molecule, which then gets turned into glucose when we're talking about glycogenolysis. Okay, so this process here has a few rules. So the first one is this chain here has to be at least 11 long. Okay. The reason it has to be 11 long is because you need, the minimum it can take is 7, and you also need these 1, 2, 3, 4 spaces here in order to allow other enzymes to attach, okay? So the minimum it can ever take is 7, okay? So 7 is, rule number 1 is there's got to be 7 minimum. Rule number 2 is that when it branches, they must be spaced at least 4 residues apart. And the third one, an obvious one, is that when it branches, it has to go back to a point before, closer to the glycogen in the middle, okay? Because that way it's going to fill up space. So it's towards, oh, my handwriting's awful again, towards glycogenin, okay? The glycogenin plays another important role because let's say we go through full glycogenolysis, and basically what we will end up with then is, I'll leave these rules on the board for now so you can remember them. We go through full glycogenolysis and we end up with and this is relating to glycogen synthase as well, why that's important. We end up with our starting molecule, the fundamental building block of glycogen. Now, glycogen synthase can't act on anything less than 8, it's less than 7, okay? Or less than 8, which is exactly why there's got to be a minimum of 7 before that branch, so the glycogen synthase can work along that branch, okay, to expand the glycogen. So glycogenin plays a massively important role because it plays an important part in the synthesis of these first, I think it's 8, 1, 2, 3, 4, 5, 7, 8, yeah. I think it's 7 or 8, these first 7 or 8 glycosyl residues, okay? So that's why glycogenin is massively important. And what's interesting about glycogenin is these are sort of 2 matched, sort of twin proteins, if you like. this side here is responsible for producing these residues, and this side here is responsible for producing these residues here. So now we have a complete understanding how from this we can build up glycogen for storage of glucose as the form of glucosal residues. And now we also have from the previous video how that breaks back down again to this raw centre part in a glycogen granule. So the final pathway we're going to talk about now, we're going to the final video, is